Okay, so welcome to this next video in which we are discussing atherosclerosis. Okay, so we're currently in the process of discussing some prerequisites for understanding the pathogenesis of atherosclerosis. Okay, so we've studied the structure of the arterial wall, and now we're in the process of discussing the structure of LDL molecules, and to do this I'm discussing with you the different types of lipid molecule. Okay, so we're starting off by discussing what phospholipids are, and we've seen this major example of phospholipids, which are phosphoglycerolipids. Okay, we're about to now embark on a discussion of another type of phospholipid, which are the sphingomyelins. Okay, right, so let me get another piece of paper for this. Right, so sphingomyelins are a subset of a larger group of lipid molecules known as sphingolipids. Okay, so let's start off by discussing what sphingolipids are, and then we'll see that sphingomyelins are just a subset of the sphingolipids. Okay, so sphingolipids are all derived from structures known as ceramides, and ceramides are all based on the structure of sphingosine. Okay, so we need to start off by discussing the structure of sphingosine. Okay, so sphingosine is what we're going to start with. Now, sphingosine is again the old biochemist's name for a molecule that is now more correctly called 2-amino 4-octadecene 1,3-diol. Okay, so 2-amino 4-octadecene 1,3-diol. Okay, and although this is a real mouthful, it's a useful name because actually it tells you exactly what to do. Okay, this is not complicated. You can read this without too much effort. This bit tells you we have an 18-carbon molecule. The ene tells us that we have a double bond. Okay, now where do we have the double bond? Oh, coming off the 4th carbon. Okay, then off the 1st and the 3rd carbon we have alcohol groups, that's the 1,3-diol back here, and then off the 2nd carbon we have an amino group. So that's very, very simple. All that's interesting is the 1st, the 2nd, the 3rd, the 4th, and then also the 5th carbon, because the double bond goes between the 4th and the 5th carbon. The other 13 carbons must just be completely boring and saturated. So let's draw out the first 5, and then we'll abbreviate the other 13. Okay, so 1, 2, 3, 4, 5. Those are the first 5. Then we'll have 12 methylene groups because there's nothing interesting after the 5th carbon where the double bond ends. Okay, so here are these 12 methylene groups, and then right on the end we just have a methyl group basically, and those are the last 13 carbons, 1, 2, 3, 4, 12, all the way in there. Okay, and then 13 is over here, and then the other 5 there to take us up to 18. Right, so let's now discuss the interesting bits. So we have a double bond here between the 4th and the 5th carbons here. Okay, and those will also have hydrogens coming off. One off the 4th and one off the 5th. Okay, so that's the 4-octadecene bit. Completely dealt with now. So we've completely sorted this bit out now. Okay, this is taken care of here. Okay, and also with those 3 carbons to take us up to 18, of course, but we'll keep those separate. Okay, right. Now let's handle some of these other ones. So we'll start off with 1,3-diol. Okay, so we need an alcohol group coming off the 1st carbon here, and also the 3rd carbon here. Okay, so that's the 1,3-diol completely taken care of. And let me just straighten up this piece of paper. Okay, that's better. Right, so here is the alcohol group coming off the 3rd carbon, and here is the alcohol group coming off the 1st carbon there. Okay, and now I've only got one last thing to deal with now, which is the 2-amino here. So we need an amino group coming off the 2nd carbon. Okay, so let's just add that in now. So here's a nitrogen with 2 hydrogens coming off it, and that's our amino group. And now we just need to saturate the rest of the bonds coming off this carbon, well, these carbons, with hydrogens. Okay, like so. And then we have our structure for sphingosine, or 2-amino 4-octadeciane 1,3-diol complete. Okay, so this is the structure of sphingosine. Now, the reason I've shown you this is that we have these very important groups, these alcohol groups that come off the first and the third carbon, and we have the amino group that comes off the second carbon here. And then we have this extremely long hydrophobic tail here. Okay, from now on, however, I'm going to abbreviate this structure down, and now what we're going to show it as is just one single line, the way we abbreviated down the long-chain carboxylic acids down to a line, basically. So now this line represents sphingosine, and this represents my really long hydrophobic tail here of the sphingosine molecule. Okay, so this is sphingosine. Now let's discuss how we can turn sphingosine into a molecule known as a ceramide. Okay, so let me discuss how you create a ceramide from sphingosine then. So basically, to make a ceramide from sphingosine, what you need to do is attach a long-chain carboxylic acid onto the sphingosine molecule, and the way you attach it on is you bind it to the amino group. The clue's in the name, amide. So we could have attached it onto these two alcohol groups here, but no, we're going to attach it onto the amino group. So let me just remind you of an amide link, because it would seem unfair for me not to do this when I've discussed esterification and binding phosphate groups to alcohol groups as well. I don't know whether or not that's called phosphoesterification. It would make sense. Okay, so now let's talk about the formation of amide links. Okay, so we've got our amino group here that comes off the second carbon of sphingosine. Okay, and then we've got our long-chain carboxylic acid molecule here. And here's the carboxylic acid group, and then here's the arbitrary hydrophobic tail. Okay, and then what we're going to do, of course, is break the alcohol group off the alpha carbon. Oh, sorry, not the alpha carbon, off the carboxylic acid carbon here. Okay, and we'll bind that to the hydrogen that will break off this nitrogen here to make water. Okay, so it's a condensation reaction as always. And then we'll bind this nitrogen atom here of the amino group to the carbon of the carboxylic acid group, and this link here is known as an amide bond, okay, or an amide link. So this is how we're going to attach our long-chain carboxylic acid onto our sphingosine molecule. Okay, so I will draw this like so. So I have a little line coming up here which represents our amino group, and then we've got this long-chain carboxylic acid here. Okay, and I'll just denote this as Fa, which I agreed with you earlier, stands for fatty acid, the other name for a long-chain carboxylic acid. Okay, so this then is the structure of a ceramide. Now, a ceramide then is not just one molecule. Sphingosine is a single molecule. Of course, we're not taking into account all the stereoisomers. But if you ignore the stereoisomers, sphingosine is one molecule. Okay? Ceramides are not just one molecule. They're a whole family of molecules, because it's not set in stone which long-chain carboxylic acid you are going to attach onto the amino group of the sphingosine molecule here. There are many different fatty acids you could attach there, and therefore there are many different ceramides that you could make. Now, sphingolipids are then a further modification of a ceramide. So you take some ceramide, okay? And if you want to turn it into a sphingolipid, what you're going to do is you're now going to attach a group onto this alcohol group here. So all you have to do is attach some sort of group onto the alcohol group that comes off the first carbon of the sphingosine molecule, and then you've got a sphingolipid. Okay, so you just need to attach some group here. Now, that obviously has a huge scope for variation. We're going to narrow down the group that you add on here somewhat. Okay, and that will give us the category of sphingolipids known as the sphingomyelins. Okay, so this is the general sphingolipid, where you attach an arbitrary group onto the alcohol group of the first carbon of the sphingosine molecule within the ceramide. Okay, we are now going to talk about a subcategory or a subset of the sphingolipid molecules, which are sphingomyelins. Okay, now, in the sphingomyelins, this group that you attach onto the alcohol group of the first carbon of the sphingosine molecule is more restricted. Okay, so it needs to contain a phosphate group. So let me show this. So we'll draw out our ceramide structure again. So here is our sphingosine molecule with the long chain carboxylic acid attached to that amino group coming off the second carbon via an amide link. Okay, and here is the sphingosine molecule. And now, coming off the alcohol group of the first carbon of the sphingosine molecule, you are going to have a phosphate group. Okay, always in sphingomyelins. So you'll take your phosphate group and you'll attach it to the alcohol group of the first carbon of the sphingosine molecule via a phosphoester link. Okay, so this is a phosphate group here. Okay, and then you go further, because remember, of course, you'll have the other alcohol group on the other side of the phosphate group, just like we did in phosphoglycerolipids, and you can attach that then onto another group. Okay, and an example again would be choline. Okay, so you can attach it onto another alcohol group and attach another group there, and that is the general sphingomyelin. And an example then of a sphingomyelin would be when this group that you've attached onto the phosphate group coming off the first carbon's alcohol group of the sphingosine molecule is choline. Okay, but there are other groups you can attach there as well. For instance, ethanolamine you can attach. Another example would be glycerol potentially. So there are other examples of sphingomyelins. Okay, right, and these satisfy the conditions of phospholipids as well. They have a long chain carboxylic acid within their structure, and they also have the phosphate group here, and that's all that you need in order to be considered a phospholipid. Okay, so these meet the criterion to be considered phospholipids. Okay, now, basically, when people talk then about phospholipids, they want to encompass both phosphoglycerolipids and sphingomyelins, because actually both of them are used within phospholipid bilayers. Sphingomyelins, as the name implies, are specifically extremely important in myelin, which lines neurons' axons. But you do find sphingomyelins in the phospholipid bilayer around cells. Okay, they're not as present as the phosphoglycerolipids, but they are still a considerable presence. So we like to generalize and have a picture that can refer to both phospholipids of the sphingomyelin type and also phospholipids of the phosphoglycerolipid type. So, what we often draw is this picture like so. Okay, for a phospholipid. Now, why does this make sense? Well, basically, these two lines here represent the two hydrophobic tails, okay? And both phosphoglycerolipids and sphingomyelins both have two hydrophobic tails, just like so. In the case of the sphingomyelins, you have one which comes from a fatty acid and one from the sphingosine molecule. And in the case of the phosphoglycerolipids, they both come from long-chain carboxylic acids. Okay, so these are the two hydrophobic tails of a phospholipid that, no matter whether it's a phosphoglycerolipid or a sphingomyelin, you have. Okay, now this other structure here, this represents the polar head of the phospholipid. Okay, so this is what's known as the polar head. And again, both phosphoglycerolipids and sphingomyelins have this. So in the case of the sphingomyelin, this would be the phosphate group, along with potentially the group that then comes off the phosphate group. Now, why is this polar? Well, remember, the phosphate group has that negatively charged oxygen that is singly bound to the phosphorus atom at the center of the phosphate group. That is a very polar structure because that's got a full negative charge on it. In addition, choline choline was a charged molecule. It had that positive charge coming off the nitrogen. So if you do have choline attached there, that's certainly got a polar group as well. So this structure is very polar and can interact with water well. Okay. Similarly, if we look at the phosphoglycerolipids, we've got the same thing. A phosphate group, which will have this negative charge that I was talking about here. And then also an additional group that could potentially also have a positive charge within it. Okay, so that's what is represented by this polar head here. So this is the diagram that people use to sum up both sphingomyelins and phosphoglycerolipids as phospholipids. Okay, right, so that was an extremely long sidetrack in order to explain this cartoon for a phospholipid. Okay, right, so now back to the structure then of LDL. Okay, so I said that LDL particles were these spheres of lipids, okay, and that the outer surface was made up of a phospholipid monolayer. Okay, so what you have is the heads of the phospholipids all pointing outwards like so, okay, towards the water. And this makes sense because we're going to chuck these things into the blood, so they're going to be surrounded by water. So the polar heads are nicely lining the outside of the low density lipoprotein particle here, and they'll be interacting with water. Okay, so let me colour these in. So here are the polar heads of these phospholipids, like so, pointing out towards the water. And then we've got the hydrophobic tails pointing in towards the centre of the LDL particle. And these will be interacting with the other very hydrophobic molecules that are at the centre of the low-density lipoprotein particle. Okay, so this is what's known as the phospholipid monolayer. Okay, now, embedded in the phospholipid monolayer of LDL particles, you have one enormous protein. Now, the reason lipoproteins are called lipoproteins is that they contain both lipid molecules and also protein molecules. Now, LDL particles are not massive. Okay, they've got a diameter generally of around 20 to 25 nanometers. Okay, so they're not that massive. But they are totally dominated by an enormous protein. Okay, and this is odd for lipoproteins. Generally, they have more than one protein within their phospholipid monolayer, but LDL particles only have this single giant of a protein within their phospholipid monolayer. And this single giant of a protein is called apolipoprotein B100. Okay, so all of the proteins which are present within the phospholipid monolayers of lipoproteins are called apolipoproteins. And the reason for that is the lipoprotein refers to the entire particle, the mixture of lipids and protein. Okay, apolipoprotein refers to the protein without the lipid. Okay, so this is called apolipoprotein B100, and it is a giant compared to some of the other apolipoproteins which you find in other examples of lipoprotein particles. Okay, right, so it sits within the phospholipid monolayer. So let me now complete the phospholipid monolayer here by just making up the remainder with phospholipids. Okay, so here are the polar heads then in turquoise, like so. Okay, right, so that's the outer surface then of the LDL particle. You've got this phospholipid monolayer with the apolipoprotein B100 within it. The next fundamental component of LDL molecules that we need to discuss is cholesterol and also cholesterol esters. But I think we'll do that in the next video.